what is detected in MRI, the relaxation or the resonance of those different atoms. And the strength of the signal depends on the chemical composition of the tissue. So if you're using a sequence that's specifically for oxygenated blood, which is what MRI does, it's going to depend on the concentration of oxygenated blood in an area. If you're using a signal that's sensitive to water, for example, or lipids, then it'll depend on the concentration of those different atoms in a certain volume. Okay. Does that sort of make sense to everybody? We're going to unpack a little more. So basically, the first step is the polarization, so aligning those magnets. So when you put somebody in a magnetic field, so before you're in the magnetic field, as you are now, no magnetic field, all of these, the spins of these different atoms are sort of randomly oriented, right? You put somebody in this huge magnetic field, and the external magnetic field will sort of line up the spins of the nuclei, so that the majority of these atoms will be in a given direction. Okay? So these molecules will be slightly more aligned with the field than against. Right? And then when you use your you'll excite the atom. So you'll be in a static field. You then start sending radio frequency pulses that'll sort of knock these atoms off of their B naught axis. So that axis that they're aligned. So you get a radio frequency pulse that pushes the atom, and the pulse causes the spins to sort of tilt out of the axis of the magnetic field, and over time they'll sort of relax back. They'll want to relax back to the resting magnetic field, and by varying the standard magnetic field, only spins in particular locations can be excited. So you can basically give radio frequency pulses to areas selective to different parts of the brain to sort of focally stimulate those parts of the brains and atoms in those parts of the brain to image activity in those areas. Okay. And so basically, after excitation, spins slowly realign with the magnetic field, and while they're doing this, they'll emit a radio signal. And the relaxation speed is measured by what's called T1 and T2 components. So basically, what you see here is the axis, an overhead view of that axis that we saw before, and you can see that basically the radio frequency pulse will perturb the atom. It'll slowly begin to recess back to its resting state. And this will cause a decay in the XY axis and recovery back to the Z axis. Okay. So basically, the relaxations for these T1 and T2 will help you understand properties of different tissues, and we can and different biological materials have different T1 and T2 characteristics. Okay. And so if we know the T1 and T2 characteristics of the things that we're imaging, we can have a good idea of the signals that we're getting in our neuroimaging. And so what we can do is we can then select specific slices. So in neuroimaging, we're interested in slices that are going through the head. And by varying the magnetic field along the Z-axis, only tissues in a particular location will get excited and emit a signal, right? So if we're very specific about our radio frequency pulses, we can be very specific about the molecules that we're imaging, and then we can look at how those molecules, the concentrations of those molecules within certain parts of the body. And so a lot of times people ask, well, what's the difference between MRI and fMRI? So basically you're using the exact same scanner, the only difference is the sequence that you're using. So in MRI studies, you're concerned about anatomy. You're using sequences that are sensitive to lipids or water, while functional MRI studies function of the brain, and you're looking at sequences that are sensitive to blood oxygenation. Okay? So when you're getting an MRI, the MRI scanner that you use to take images of structural images of parts of your body, is the same scanner that can be used to do functional images. The only difference is the radio frequency pulses that you're delivering to the body and to the people that are in the scanner. Okay? And so in fMRI, we're looking at activity in the brain, or our hope is to look at activity in the brain, but we're not really looking at neuronal firing directly. So you've probably seen, I think, Kathy Cullen and Ed Connor and some other people that record from neurons in the brain. They're looking at spiking of neurons. What we hope to do with fMRI is look at blood flow, which is related to spiking. So when you have some synaptic activity, it releases glutamate, which then signals your body to send oxygenated blood to certain areas to give energy to your brain for those synaptic computations. Okay? And with fMRI, what we're doing is we're not directly imaging this synaptic activity as you are with electrophysiology. Instead, what we're doing is we're going to image oxygenated blood. So we use pulse sequences that are sensitive to oxygen in blood. Okay. So the idea is that synaptic firing causes glutamate. This signals to your body that you need energy in a certain part of the brain, and more blood flows to that part of the brain. So we think that increased blood flow is related to neural computation and synaptic firing. Okay? So basically the idea of the bold signal, which is blood oxygenation, blood oxygen level dependent signal, is that synaptic transmission consumes energy and oxygen, which releases glutamate. The glutamate release causes blood flow to increase, and this causes the concentration of oxygenated hemoglobin to increase, where the blood flow increases. And our inference is that increased neural activity or synaptic transmission then leads to an increased MRI signal or bold signal. Okay? So one of the big limitations with fMRI is we're not actually directly imaging neuronal firing. We're imaging blood flow related to metabolism related to neuronal firing. Does that sort of make sense to everybody? And so you can basically, with bold signaling, another limitation relative to maybe the electrophysiology techniques that you saw before is that you're really limited in terms of temporal resolution. You can only acquire an image of the entire brain approximately every 1 to 4 seconds, right? So you're very limited in the temporal resolution that you have. But at the same time, you get pretty good spatial resolution for measuring over the entire brain. So, when Ed Connor or Kathy Cullen are doing their experiments and they're recording from neurons, they can only sort of focus on neurons in very specific parts of the brain. Here, we're able to image the entire brain and look at brain activity over the entire brain and then look at connectivity between different parts of the brain, although we're limited by our temporal resolution when we do that. So again, as I said before, it's a blood flow-based signal. As neural activity goes up, local blood flow, we think, increases, and this changes the amount of oxygen and hemoglobin in an area. We're imaging the oxygen and hemoglobin in an area, and this is what gives us the fMRI signal that we're looking for. Okay? Is that kind of sense to everybody? And so, as I said before, the temporal resolution is really slow. And the reason it's really slow is your blood flow to a certain area is lagged relative to the neuronal firing that you see, right? So here you see time in seconds, from 0 to 25 seconds, and then this is the hemodynamic response function. So basically, the intensity of that oxygenated blood signal. So you could think of showing somebody a cue at experiment stimulus onset here, and then neuronal firing is going to happen really quickly after they see that cue within the brain. But the blood flow to that area of the brain is actually quite lagged. So this is what the blood flow response function looks like for a given experiment stimulus onset. So you can see that the peak bold response is actually delayed by like about 6 seconds. So you're not able to get very precise timing on what is happening in the brain relative to stimulus onset with neuroimaging. So you have to do some tricks to sort of model this lag. And the thing to realize is the lag is different for different parts of the brain. Right? So you have a different hemodynamic response function. This this hemodynamic response function, although we assume it's the same over all parts of the brain when we're doing our neuroimaging analysis, it's actually quite different in different parts of the brain. Okay? So there are big assumptions that are made about what this response function looks like. Okay? Does that kind of make sense? So important aspects of the Bolt signal. Because Bolt signal is based on blood flow, the temporal resolution is limited by the precision of blood flow regulations. This is in contrast to electrophysiological studies in which you have really fast recording of neuronal firing. The spatial resolution is largely limited by the strength of the signal. So you're imaging voxels in the brain, so volume elements in the brain that are about 1 millimeter by 1 millimeter if you're imaging with a 3D scanner. And if you think about it, there are hundreds of thousands of neurons in each voxel. So when you're doing those electrophysiological recordings, you're pretty sure you're recording from single neurons. But here, when you see those blobs in the brain, those are hundreds upon hundreds of thousands of neurons that are responding in response to a stimulus. So it's really hard to know selectively what different neurons are doing with every neuron. And the other thing is you can't measure absolute amounts of activity or metabolism. You can only measure differences between two conditions. So fMRI signals are always relative to sort of a baseline. And we'll sort of go into that when I explain some of the experiments, procedures that and as I mentioned before, every region of the brain has a unique hemodynamic response function. So we generally assume that the same hemodynamic response function over the entire brain, but different areas have different response functions depending on vascularization and sort of speed of processing. Does that sort of make sense related to the limitations? Right, so as I mentioned before, more limitations, because there are a lot of them for fMRI. It's sort of a population-based method. We're not getting single neuron reporting. We're getting hundreds of thousands of neurons when we're looking at a particular imaging blob. It measures synaptic activity, so input and local processing and not spiking output. So remember, it's measuring metabolics of certain regions of the brain and not actual spiking output. You have coarse temporal resolution, as I said before, and it's easy to use, but it's also easy to misuse. So basically what you can do, what a lot of people do, and a lot of those New York Times articles I mentioned before do, is they sort of just throw somebody an MRI scanner. There are a lot of statistical packages that allow you to very easily analyze MRI data, right? But because of these limitations, a lot of their data is very hard to interpret. So it's very important to sort of understand the limitations of fMRI to know what you can and cannot reasonably say about what's happening. Okay? We're going to go over some of those limitations in a second. And so the logic of, so does anybody have any questions about MRI experiments or just like basic physics stuff? So the logic for fMRI experiments is basically to present a stimulus or have the subject perform a task in a stimulus and compare it to some rest image. Okay? To measure the bold signal, you'll measure bold signal to find regions of the brain showing increased activity to stimulus or tasks as compared to a control condition. So you're doing differences in brain activity relative to some rest conditions or some control conditions. And so to analyze fMRI data, what you generally do is you partition the brain into voxels, and you're basically looking at one measurement over time for each voxel in the brain, and you're sort of pegging those to different task conditions throughout your experiment. And then what you can do is use pretty standard statistical tests to regress and figure out how task conditions regress onto neural activity. We'll go over some of the general linear models that we've used to do those sorts of analysis. One of the main problems with MRI imaging is you're partitioning the brain into hundreds of voxels, and so you have multiple comparisons. So the statistical tests that you're doing, you need to make sure that you correct for the fact that you're looking over comparisons over hundreds of different voxels to make sure that the results that you have are statistical. And so the overview of an fMRI experiment is basically put somebody into the MRI scanner, you image the entire brain and partition the brain into a series of voxels. So you can partition the brain into hundreds of voxels over the entire volume of the brain. And for each of these voxels, then, you can generate a time course that measures bold signal through the time course of your experiment. Okay. You can then do basically a linear regression where you take the bold signal and you look at different task conditions, which are represented by X1, X2, and regress, do some regression where these betas represent how much variance in this neural signal is accounted for by your different task conditions. And since you're doing this over the entire brain, these time courses over the entire brain, this general linear model is over the entire brain, you can then map these beta weights to different parts of the brain to see how different brain activity responds to different task conditions. Okay? Does that sort of make sense to everybody? So you're basically doing a general linear model or regression between neural activity and different task conditions. Find these regression weights, these betas, and mapping them on to different parts of the brain to figure out how they're related to different task conditions. Okay? And so basically, the equation for a single and all voxels is given by a general linear model that looks like this. You have your data for a given scan, indexed by J. So you have all your scans through the time course of an experiment. And then you can basically just fit, for each of those scans, the different task conditions that you have, and find the beta weights, basically how much variance is assigned to each of these betas for a different task condition relative to that time course. And so the matrix formulation looks something like this. You have Y, which is your image, your different betas for different task conditions. You do this over all your scans. So from the first scan through the J scan, so basically that's the full time course of your experiment. And then you regress to find the beta weights relative to your experimental conditions. Okay. So the way this looks, you can think of this experiment here. This is sort of an example that describes what's going on. It's pretty simple. So the idea is that you've got rest conditions in light gray. You have conditions where you're shown a visual stimulus here. So basically here we're trying to look at brain activity related to presentation of a visual stimulus. So there's a rest condition. The blue regions represent presentation of the stimulus. And then the dark gray regions represent scrambled objects. Basically the same stimulus but scrambled so that you match all the properties of the image, except the features of the image itself, like its form. Okay? So you can basically then create a general linear model where you take the MRI signal from a given voxel, and then you have conditions for the scrambled image, the intact image, and you basically regress on them to get your betas. So you're basically asking, in your data, what can we explain using our design matrix, and how much of it can we explain using the beta weights associated with your different task conditions. So statistical significance is then basically based on these betas. Okay? If they're higher, it means things are more active or more statistically significantly active for different task conditions. So in the example of the visual stimuli I showed before, you can basically look at contrast between the regression coefficients for intact versus scrambled image, and you might see some activity in visual cortex because visual cortex is going to respond to presentation of visual images. And you can also, so you can do that relative to baseline. You can also do that relative to a different task condition. Right? So one of the limitations of looking at an image relative to baseline is if you have somebody just looking at a blank screen, you don't know if the activity that you're getting is related to just presentation of object by itself, relative to a blank screen, features of the object. And so a better control is to use the scrambled images that we saw before, so that you're sort of controlling for all of the fundamental components of the image and not showing the features themselves. Right? So does anybody have any questions so far? So basically, now I thought we'd go into ways to sort of critically evaluate fMRI. So a lot of times you see in the newspaper claims like this, that in this experiment, this is cerebellum. You might have learned that in some other lectures, and people might say, well, region X, cerebellum is activated when subjects do Y. So here, this study was done, and people said the cerebellum is activated when people look at faces. Okay, and basically the contrast that people did was they showed faces to subjects and had them also look at a blank screen, did a contrast between those two conditions, and found that faces were activated. This is a bit strange because the cerebellum isn't really known to be face selective, right? It doesn't usually activate for faces. So you have to think about, when you see claims like this, you have to think about what is it compared to, right? So when I'm comparing to baseline, there are a lot of, and just a blank screen, there are a lot of things that people can be doing, right? And one of the things that could be happening is, the reason you get more activity in cerebellum relative to, for faces relative to baseline is because eye movement is happening. So you might have learned in Captain Cullen's lecture that cerebellum is actually responsible for eye movements, right? So these signals that we get, we don't know that they're really face selective because you haven't done a control relative to faces. Instead, they could be related to eye movements, for example, right? And so you need to make sure that the controls that you're doing when you're doing these fMRI experiments are well thought through. And you understand what the conditions that you're comparing to are being used. Okay? So in fMRI, you can only tell about the difference between conditions and not absolute activity. And so you need to determine the control condition and think about all things that differ between the test condition and the control condition. So in this case, one thing that could differ is this idea of eye movements and how that could activate cerebellum, which isn't really a face selective area, but activates just because of eye movement in the face condition. Okay? Does that make sense? Another claim that is often made is that when people do mental task X, they activate region Y, which is known to be involved in the mental process Z. Therefore, X invokes process Z. All right, so that's a very complicated way of saying something that you see here. So an example that's commonly given is watching someone else in pain activates brain regions known to be involved in experiencing pain. So empathy is feeling another person's pain. Okay. And so what was done in this experiment was what was done in previous experiments is if you put somebody in the scan, temporal gyrus insula activates in that painful condition. And so then what another set of experimenters did was they went in and they said, well, we're going to look at empathy. We think empathy recruits the same areas as pain. So we're going to put people in the scanner and we're going to have them look at previous romantic partners that have done both, right? And this is going to then activate, this will activate pain. This activates pain. Okay? So people have done this with romantic partners. They've also done this watching somebody else get hurt. And you routinely find these areas of insula and temporal zones, right? I mean, that sounds hokey, right? But these things are actually published in really good places. So this is actually published in Science. The thing you have to realize, though, when you're doing this is you're relying on what's called reverse inference. Okay, you're basically inferring the function of a brain area without actually testing its specificity for that function. Right? So you can't really make those claims unless you directly test them. So one way, the better way to have done this experiment is to sort of go towards converging evidence. Okay? So one thing that could have been done that would have been more convincing would be to have people look at these empathetic situations, looking at other people getting hurt, activate this area of temporal gyrus insula, also have them experience physical pain, and see if actually the same brain regions are activated when they experience pain itself and when they see people experience pain, or they are, looking at pictures of their previous romantic partners, right? Things that you think will cause emotional pain. Right? So if you do that, then you have sort of converging evidence that the same sets of neurons might be involved in these two functions. It still doesn't rule out, though, task confounds, right? Because you need to think about the task that people are doing. So when you're simply viewing images of people getting hurt or previous romantic partners, there's a lot of things happening in your brain, right? Besides thinking about pain, right? There could be signals related to the surprise of seeing somebody previously new. There could be surprise signals related to what are called prediction errors, so like uncertainty about events occurring. And those signals, we actually know, recruit this same region of the brain. So there's still questions, even if you do the thing where you have them do both the actual physical pain task and the empathy task, we still need to have a really well-controlled experimental design where we rule out other factors related to what people are thinking about when they're doing the task in the scanner itself. And so then you have to really ask, is it really the same region that is activated in those studies, right? And so back in the day, so this was 2004, you could kind of get away with this kind of reverse inference. Nowadays it's a little bit more difficult, but still you see sort of creep into imaging studies. So that actually kind of concludes what I had for today. And tomorrow or Friday we'll talk about experiments sort of using some of these ideas. I'm happy to take any questions if you have them. And yeah, that's all I have. Oh yeah, feel free to clap. Yeah.